My pleasure to introduce Buddy Weissman. I've known Buddy for 20, 25 years. We met when we were 20.
That would be a lot harder.
Yeah, absolutely. Actually, he sat on Pathology B, you know, knows that study section, a long time ago. We were just reminiscing. We used to get 160 grant applications. They were all paper. We used to argue about the budget. So be glad you're where you are when you are. So he's a professor of pathology and laboratory medicine at the University of North Carolina. He's a member of the Library Cancer Center, Comprehensive Cancer Center, and he has a distinguished research program, as you'll see. And he's used a combination of somatic cell genetics, positional cloning, mouse models, and other technologies. And was really at the forefront of coming up with the concept of the tumor suppressor gene. Sounds pretty simple now, but it wasn't then. And he's also was the forefront of identifying metastasis suppressor genes. And when those first came out, they were very controversial. And people said, no such thing as a metastasis suppressor gene. Well, as it turns out now, there are many metastasis suppressor genes. The first one, he told me this morning, it's called KISS, K-I-S-S. He did this work with Danny Welsh, and Danny was at Hershey's. So that's why that was called KISS, systematic nomenclature. But this is another example of the importance of staying in research long enough to turn all your critics into competitors. And that's what happens. So it's kind of the nature of the business. So he's been focused on chromatin remodeling, which is a very complicated topic, and how it contributes to cancer development and malignant progression. And he's studying how chromatin instability can silence gene expression by a mechanism that is independent of methylation. So it's just amazing. So when I invited him, he was glad to visit. His concern was snow. We don't have any. When I went into the lobby to pick him up, he had his back to me, and I could tell it was him because he had a little snow cap on, and winter jacket from North Carolina. The only place he sees snow is on the Weather Channel. Buddy, that's enough of me. Welcome. Thank you so much for
Well, thank you very much for the invitation. I've been here twice, but only in the airport, so this is the first time we've actually seen some of the city, and it's obviously very beautiful. What I'm going to talk about today is our long-standing, our 20-year foray into the field of cancer epigenetics, and specifically on chromatin remodeling complexes, which, as Jim said, now is like a very hot topic, and there are lots of competitors. But I think it's a really interesting question both for genetics, pathology, and I'll try to touch on some of those.
no luck.
That won't advance, it's just the laser.
Oh, all right. Well, then I can do this. All right, so I'm going to talk about, I'll give you some background about chromatin remodeling in cancer, and then I'm going to talk about what I think are some of the key questions that are facing us in this particular area of research. One of them being, how do mutations in this chromatin remodeling complex cause cancer development? I'm going to talk a little bit about some of our translational studies where we've identified some therapeutic vulnerabilities, and then I'm going to finish up a little bit about tumor specificity. All right, so I didn't put this in intentionally. Whoops. I didn't know if I had a financial disclosure, but considering the breakfast you're having here, it's turned out to be very apropos. Oops. Okay. So this is the problem we all face as eukaryotes, trying to put approximately 3 microns, meters of DNA into a 3-micron nucleus. And eukaryotes have evolved a very complicated mechanism of folding and wrapping and coiling to actually accomplish this task. This is, with the basic unit being a histone with approximately 167 base pairs of DNA wrapped around it. As you can imagine, with such tight packaging, this is an inhibitory
I think the battery died.
To all the types of basic cellular machinery, including transcription. And therefore, we have evolved several different mechanisms to overcome this problem. The ones that you probably have heard a lot about are histone modifications. There's also different types of histones. And what I'm going to be talking about are the ATP-dependent chromatin remodelers. There we go. I guess I can. No. All right, I'll make do with this.
This works fine.
Okay. So, just to be, in all fairness, there are 4 different chromatin remodeler families. And they all have different functions. Most of these are actually more involved with condensation of nucleosomes. We're going to be talking mainly about the SWUSNF complex, which is involved in actually repositioning nucleosomes and often to allow more space between them. So this is a complex that was initially identified in yeast, and it's called SWUSNF because in yeast, when you knock out any member of the complex, you find that you inhibit the mating type switch in yeast, and they also become sucrose non-fermenters. It's an ATP-dependent complex, which will become an important point in a moment. When you knock it out, you affect the expression of approximately 6% of all yeast genes. It's multi-subunit, anywhere from 9 to 12, obviously very large. And what was interesting in yeast is that basically knocking out any subunit of the complex gave pretty much the same phenotype. It's highly conserved. There's about a 70% amino acid conservation between yeast and humans, and the smallest subunit, SNF5, is on the amino acid level identical between mouse and human. So it's clearly a very important complex. We have a nomenclature issue with this, and I unfortunately will often go back and forth because when I got into the field we were using these, and then later the group in England officially named them these, and I can't even tell you what this acronym is anymore. But even some of the subunits don't actually follow those acronyms. So it's definitely confusing, and you'll see papers that use both of these. And in fact, SNF5, which is this gene, it's not even up here, and it has a 4th name also. So forgive me if I go back and forth. I'm mainly going to be talking about this subunit and this subunit, which I'll call SNF5 for most of the slides. So what's so important about nucleosome positioning? Well, I like this analogy. Think of putting a nucleosome as a comma in a sentence. So this is a very different meaning than this sentence, and this has a really different meaning than this sentence. So where you put nucleosomes is very important for how the epigenome and transcription and DNA repair and DNA replication occurs. I'd like to point out that we have somewhere around 485 different histone-modifying genes, and this was a few years ago. It wouldn't surprise me if there's more by now, because there's new modifications that are found, have been found since then. And and if you look in cancer, there are a variety of different mutations that occur through all these different, a large number of these different histone-modifying enzymes. In contrast, there's really only the limited number of complex members that I showed on the previous slide. And what we think is that the diversity that you see in complex function is basically determined by which of these alternative subunits are present in the complex. I should also point out that the complex, as I said, is driven by an ATPase component, either BRG1 SMARCA4 or BRM SMARCA2, And they are mutually exclusive. So complexes have either one or the other ATPase, and you can further kind of divide these into two other types of complexes where either the ARID1A, ARID1B are present with SMARCA4, or the PBRM ARID2 combination is present with SMARCA4, but these never occur with SMARCA2. How did we get this connected to cancer? Well, in 1998, through old-fashioned brute positional cloning, Oliver Delatre's group found that a very rare tumor, malignant rhabdoid tumor, and this is the pathology department, and I'll point out it's not a rhabdomyosarcoma, it's a much rarer tumor, that virtually every case of these cancers had a truncating or inactivating mutation in the smallest subunit of the SWUSNF complex SNF5, the other name is INI1, or SMARCB1. And since that time, and actually this is a slide I put together in April, so I'm sure it's like this now, the field has just exploded in the number of publications. And one of the reasons is that a recent paper from, 2013 paper from Kadosh and Jerry Crabtree's lab showed that if you look through all the TCGA data, that approximately 20% of all human tumors have a mutation in at least one member of the SWUSNF complex, in some ways making it the most frequently mutated group of proteins in human cancers. And one of the questions which we'll come back to is, interestingly, unlike RB, which is mutated in a variety of different types of cancer, P53, you name your favorite tumor suppressors, these genes tend to be mutated in very specific patterns. So SNF5 occurs in malignant rhabdoid tumors, 50% of which are in the kidney, 30% of which are in the brain, and which are now called ATRTs. SMARCA4 is mutated in about 10% of lung adenocarcinomas, but not in lung squamous cell carcinomas, and the tumor which we'll be spending some time on. Small cell carcinoma, the ovary hypercalcemic type. ARID2 is in hepatocellular carcinomas. This subunit turns out to the fusion protein SS18SYT is the driver for synovial sarcoma. Recent work from, again, Crabtree's lab showed that this subunit knocks SNF5 out of the complex and actually is a gain of function for the SWUSNF complex. So there's these very specific tumors that are driven by very specific mutations. And we actually don't understand at this point what the mechanism is for that, although I have some ideas. So we're going to talk for most of the talk now about this rare tumor, which rather than saying this long name, we call SCOOT. Okay, so this is a relatively new tumor. It was first described in 1979. As you can see, it's extremely rare worldwide, less than 500 cases reported. 3 or 4 years ago, I'd say we probably have been able to dig up another 200 or so since, because these are often misdiagnosed. They generally occur in young women, however, they can occur in children, and I think this may be a little bit older, but they are never seen in postmenopausal women, which is an interesting point. It's called hypercalcemic type because 6% of the cases actually have hypercalcemia. Other than that, and the reason I think it's often misdiagnosed is that it has symptoms very similar to other types of ovarian cancers. It's rather large at initial treatment, and we really don't have any standard care for this. Basically, there's so few cases, they remove the tumor. It initially responds really well to chemotherapy and radiation. With 6 months, these return, and they have a very poor survival rate. I'm not sure there are any patients that have survived more than up to 5 years. There are also unusual tumors, and this is actually similar to malignant rhabdoid tumors. There's very little evidence of any genomic instability, very few copy number changes. Generally what you see are only changes around the site of the SMARCA4 gene, which is on the short arm of chromosome 19. And we're increasing these numbers now, but everything we looked at so far shows that they, in terms of their gene expression profile, are distinct from the more common serous cell carcinomas, ovarian carcinomas. So four different groups went ahead and got enough tumors to do whole exome sequencing, and in 2014, 4 papers came out. 3 of them were back to back to back in Nature Genetics, and one in Rare, and one, actually the original group was a Polish group, so it was not well recognized initially. And what they showed was that virtually 99% of these tumors all had mutations in SMARCA4. And like SNF5 mutation in rhabdoid tumors, the vast majority of them were nonsense mutations, splice site mutations, and only a few missense mutations. And there's generally like one hotspot here, and this is one that also occurs frequently in lung adenocarcinomas. You'll also notice that there's not a particular hotspot here, although clearly a lot of these tend to fall towards the C-terminal end of the protein. And so as of 2014, it was about 94% had mutations, and then when you actually looked at cases on tissue arrays, it still held about 94%. So this clearly is a key loss. We've shown that actually not only are they missing the SMARCA4 ATPase subunit, but they also lack expression of the other ATPase subunit, both in primary tumors, and this is SNF5 as a positive control in a sense. There's only 3 cell lines available, which of course limits work and really increases the competition. But both of these cell lines have SMARCA4 inactivating mutations, and SMARCA2 is missing. In all these cases, the SMARCA2 is epigenetically silenced, and we can reactivate it, such as with an HDAC inhibitor. And it's interesting to point out that the rhabdoid tumors, and this is some of our earlier work, also, these are all rhabdoid cell lines, they all lack expression of SMARCA2, again, epigenetically silenced, while a variety of other types of pediatric cancers are positive. So this obviously has some importance. And I'll also show you that using the TCGA data from a study that we published in 2014, although we didn't include this data, we were able to show that in these tumors that had very low BRG1 mutation or had known inactivating mutations, that SMARCA2 expression was also low compared to all the BRG1 wild-type tumors. So there seems to be some association between loss of BRG1 and silencing of the other ATPase subunit. I'll point out also, and I want to stress that if you look in the TCGA data, there's somewhere around 500 SMARCA4 mutations across human tumors. Most of them, other than this 10% of lung adenocarcinomas and the small cell and the SCOOHT tumors, are missense mutations. And there will be a paper coming out soon in Nature Structural Biology showing that those missense mutations actually do alter the function of SMARCA4, but it's not clear yet exactly what their role is in tumor development. So this isn't the world's most, this is probably the obvious hypothesis. And we already had a paper in lung adenocarcinoma showing that if we knock down SMARCA4, we increase epigenetic instability. So we believe that this is the major driver of these tumors, and it does this by causing changes in the accessibility of transcription factors to chromatin with resulting gene expression. And therefore, this is all unpublished data that we're in the process of generating. We decided to test this hypothesis by taking one of the SCOOHT cell lines, re-expressing SMARCA4, and then doing a complete characterization of the epigenome, gene expression, protein expression. So this is our experimental design. We're doing 4 biological replicates of the SPIN67 cell line, plus or minus SMARCA4. And I'm going to talk about the RNA-seq data. We have the ATAC-seq data. We're still working on that. We're generating the ChIP-seq data and the proteomics data at this point. And the idea is that once we have this data, we're going to integrate these to identify those sites that are potential driver pathways for the development of SCOOT. So this is the RNA-seq data, and I'll give a disclaimer up front that my student has done all this work and has turned out to be quite a savant for bioinformatics. And when she tells me something, I tend to take it on faith. So I understand the general outline of the data, but I can't say if you're going to ask me very specific questions about how we sequence things, I may not be able to answer. But basically, what this is showing is that this is looking at what's the major difference between the 4 groups where we re-express BRG1 and the 4 groups where we just put in a control plasmid. And what you see is that the major variable is the fact that these have re-expressed BRG1 versus the controls. So this shows that what we're looking at is what we want to see. And then this is a volcano plot, and what we're seeing is that this is a fairly benign low pass filtering of the data in terms of what we're using for our false negatives. But the majority of genes, when we re-express BRG1, go up, much less go down. The total amount is somewhere around 15%, which is a bit higher than is reported in other studies looking at SWUSNF complex members. But this is pretty much what we would expect to see, in that we expect to see chromatin opening up when we restore complex function. One of the issues that is intriguing to me is this is from our 2014 study in this lung adenocarcinoma cell line, where we knocked down BRG1 expression, and this is microarray data, but there were several key genes that we found. Obviously, we knocked down BRG1 SMARCA4, but some genes we could validate in a second knockdown cell line, and we could also validate in the TCGA data as being underexpressed in BRG1 deficient primary lung adenocarcinomas. Interestingly, these same genes are some of our top hits when we re-express BRG1 in a deficient cell line. So these genes went down when we knock it out, and they go up when we put it back in very different types of tumors. And it does raise the question as to whether there are actually consensus targets that SWUSNF complex controls in a variety of tissues, and if so, what about them makes them targets? So that's an area we're very interested in. And just to show that we're not overexpressing SMARCA4 to tremendous levels, it's down here, it's clearly up. These genes up at the top are actually greater than 300 for their log p-value, and the program can't comprehend higher than 300, so they're just stuck here at the top. So one of the things that we did was to try to get some idea of maybe where these cells are coming from. And this is an important issue. We actually don't know the cell of origin from looking at rhabdoid tumors, and we really don't know the cell of origin for these SCOOT tumors. And one of the biggest complications from this is that we have floxed mouse models of SMARCA4 and of SNF5, and the question is, what tissue do you knock these genes out in order to try to create a tumor model? So this is something that is coming out soon. It actually hasn't been published yet, but this is a group that has done RNA-seq on all these different types of normal tissues. Obviously, they're much more interested in lymphoid, myeloid, stromal cells, and stem cells, and then all the rest other uninteresting cells. But the program they set up will allow you to take your RNA-seq data and ask which of these tissue types does your RNA gene expression data most closely resemble? And generally, you get not a lot of match with most tumors. So our bioinformatics collaborator was quite excited about this. So let me, first of all, this is what always confuses me. This is not a heat map. It isn't that green is going down and red is going up. This is, if it's black, there isn't much resemblance. If there's green, there's some resemblance. And if it's red, it's highly resembling. And the most interesting part here is that the cells, in the absence of BRG1, look like they're mesenchymal stem cells. They most closely resemble those, although there's, interestingly, some resemblance to Th2 positive cells. When we re-express BRG1, they now become much more like epithelial cells. So this suggests that we're actually getting a, not an EMT, but an MET, a mesenchymal epithelial transition. And this is consistent with the initial pathway analysis that we've done, where we find that a lot of the ECM reacting pathways are the ones that are upregulated when we express BRG1. So we think that perhaps these are coming from a stem cell-like population, and that when you knock out the SWUSNF complex, you're freezing them in a more aggressive mesenchymal phenotype. Now I mentioned, and we'll come back to this, there are several groups now that are actually, in the literature, are saying that SCOOT is really a malignant rhabdoid tumor of the ovary. And we had done studies a few years ago with Peter Jones looking at a malignant rhabdoid tumor in which we had inducibly re-expressed SNF5. So this is looking at the combination of all the groups, and unlike this study where we have 4 biological replicates, we only have 2 biological replicates here. But what this shows is that, first of all, the cell line identity, so here's the 2 rhabdoid tumors, plus or minus SNF5, and here's the SCOOT tumors, plus or minus BRG1, are what are basically driving the most difference between these sets of data. But it's also showing that there is some significant difference between the groups where we re-express the genes versus the groups that are controls. So this is the heat map, which is not showing up nearly as nicely as it is on my computer. But basically, as you can see, first of all, there's about a quarter of the genes that go up when you re-express, or change when you re-express SNF5 in the rhabdoids compared to what we see in the SCOOT tumors. And again, most of them go up, although there's a few in this little section here which you can't see particularly well, where they actually go down upon SNF5. But again, this is similar to the SCOOTs. When you re-express BRG1 in the rhabdoids, most of the genes go up and the smaller percentage go down. Now, this is a little more rigorous filtering of the genes, but we're using the same filter that I just showed you for the SNF5. So this reduces this down to around 4,600 genes that go up or down. And again, you see the majority of the genes go down. There are some genes that go up and not as dramatically, and then there is this group that goes down. So what we did was to compare these, and so we have 4,000 genes that are changed specifically in the SCOOTs, plus or minus BRG1. We have 747 that change in the rhabdoids, but then there's 586 genes that are in common. And when we look at those, so here are the 2 controls, here's the SNF5, here's the BRG1. In general, if these genes go up when you re-express BRG1 in either express BRG1 in the SCOOTs or SNF5 in the rhabdoid tumors, they both go in the same direction. There are a few interesting changes here, which you can't see particularly well. There's a group that goes up in the SNF5 and down in the SCOOTs.
and there's a group here that goes down in both. So we're interested to know whether what these genes are and to further characterize them from a functional viewpoint to see if there are some common targets we can use on both types of tumors. So knowing what we know, that here we have a wonderful known mutation in these tumors, is there something that we can target? And this is building upon work that came out a few years ago from Charlie Roberts and from Luong Zhang's group at Wistar, where they began looking at whether you could target the PRC2 complex on the basis of this very nice paper from Charlie's group, showing that if you knocked out the EZH2 subunit of the polycomb repressor complex 2 in a mouse model for a SNF5 loss-driven lymphoma, you totally wiped out tumor development. And they also looked in cell lines and showed that all the cell lines that were lacking SNF5 had very high levels of EZH2. So there had been a few papers that had come out that said that you could target any SWUSNF complex, any tumor with a mutation in a SWUSNF complex member by using an inhibitor of the PRC2 complex. And just to remind you, the PRC2 complex is a repressive complex. It's responsible for methylation of the H3K27, histone 3 on residue 27. It's in the histone tail. And when this becomes methylated, it causes the DNA to become, and histones become much more compacted, and is a repressive mark that's found in stem cells and many human tumors. So the EZH2 subunit actually has the methyltransferase activity, and this has become a very hot target for a variety of tumors. There are other types of cancer, subset of AML, for example, that have activating mutations of EZH2 that makes it constitutively active. So there's already been some nice clinical success with these, and as you can see, some are preclinical, but some are already in phase 1-2 trials. So we decided to look at the effect of these two inhibitors, the GSK, because anecdotally it's supposed to be more powerful than the epizyme inhibitor, and the epizyme because of where it is in clinical status. So we initially looked at cell lines and primary tumors. Got lots of pathology slides. Since I have been in the pathology department, I know what I should be presenting. So this is the level of EZH2 in, again, we don't know the cell of origin, so as a best guess, we compare normal ovary, which of course has multiple cell types, but compared to normal ovary, the SCOOTs have much higher levels of the EZH2 gene expression. Interestingly, the other members of the complex are not overexpressed, and this brings up an interesting question about complex composition. When you actually look at a variety of cell lines, and there are, as I mentioned, 3 cell lines that are bona fide SCOOT cell lines, CoA434, SCOOT1, and BIN67. There's a couple of these cell lines here that are ARID1A mutations, another subunit I mentioned earlier. Some of these have BRG1 loss that are not, SCOOT cell lines. Here's some regular serous ovarian, and then this is a rhabdoid cell line. And what you see is that even though the EZH2 protein levels aren't dramatically higher, the product, the methylation on H3K27 is much higher in all of the SCOOT cell lines, the rhabdoid cell lines, this BRG1 deficient cell line, not so much in the ARID1A deficient cell lines. And when you look by tissue array, this is the advantage of having a very strong pathology group as part of our effort. There's very strong staining of EZH2 indicating overexpression compared to normal tissue. In comparison here, it's either, 80% have high expression, and another 20% have moderate or variable expression. And finally, when you re-express SMARCA4 in all three of the SCOOT cell lines, you decrease EZH2 levels, in some cases rather dramatically. You also reduce the methylation of its target, H3K27, and this is just also seen at the expression level. So it looks very similar to rhabdoid tumors. If you knock down EZH2 in our cell lines, what you see is that you cause, and this is just an example of one of the cell lines, protein levels go down, methylation goes down, and survival goes down. So like rhabdoid cell lines, these are dependent on EZH2 expression. If you now treat with either of these inhibitors, you see very similar results in micromolar range. Even 0.1 micromolar is enough to significantly reduce the expression of the methylation. Actually, it does not change the expression of EZH2. And now this is where we see something that other groups reported somewhat differently. We do see this cell line, the HT1 cell line, in fairness, is like more sensitive to almost every drug we put on it. But all of the SCOOT cell lines are much more sensitive to the EZH2 inhibitor than the other cell lines, except, and G401 is also more sensitive than here. What's interesting is that all these other cell lines include the ARID1A deficient tumors and a few of the lung cancer cell lines that exist that are also BRG1 inactivating mutation BRM silenced. So this conflicts with the earlier data from the Zhang group. We don't have an explanation for it, but we've repeated this multiple times, and some of our collaborators have done this, and they see very similar results. And then this is just looking at the actual growth on dishes and looking at number of colonies, or excuse me, survival. What happens when we actually try this in xenograft models? We see some variation, but in both of these models we see significant inhibition of growth with the EZH2 inhibitor. Eventually there is some recovery here. This is the highest level we can use without affecting the weight of the mice. And this is similar to what's used in human patients. So it looks promising that this would be an effective treatment for SCOOT, and in fact the Epizyme phase 2 trial actually does have openings for SCOOT patients. So what is happening? We're getting both effects. When you look by cell cycle analysis, what you see is that you inhibit the S phase, when you treat with the inhibitor in all three cell lines, and after the growth arrest, you start to activate caspase, and you actually see apoptosis after a longer exposure. So these cells clearly do not like being treated with these inhibitors, and it has exactly the type of effect you would like. What's the mechanism for this? And this is where it gets a little bit interesting, or somewhat interesting. So this is the model for rhabdoid tumors, which is basically when you treat a rhabdoid cell line with an EZH2 inhibitor, or if you re-express BRG1, P16 is repressed by the PRC2 complex in rhabdoid cell lines and in primary rhabdoid tumors, and therefore you're blocking cell proliferation. If you inhibit EZH2, you reverse this mark, and you re-express P16, and the cells now become growth arrested. And actually, when you reform the complex with SNF5, it does the same thing. And after 24 hours, it knocks EZH2 off of the promoter region here. Oops, that's out. And they, I have my own. And rubber band. And they are able to, again, re-express P16. You can see P16 go up with either re-expression of SNF5 or the inhibitor. If you knock out P16, the cells don't go into growth arrest. So they're clearly dependent. In contrast, and 2 of the 3 SCOOT cell lines have very abundant levels of P16 compared to the rhabdoids where it's silenced. And when you, the one cell line that does respond, which is interestingly the most sensitive one, you actually do see the kind of response you see in rhabdoids. But these 2 cell lines are undergoing growth arrest and apoptosis, and it's clearly a P16 independent mechanism. So what do we think is happening? When we look at the morphology of these cells 5 to 7 days after treatment, or even longer, what you see is that the cells begin to look like have a neuronal phenotype. Now, I'm the skeptic in our group. I think a lot of cells tend to do this when you inhibit growth. But our collaborators were able to show that at least some of the most general markers of neuronal differentiation, MAP2 and tubulin beta 3, are also going up after treatment with the EZH2 inhibitor, and that in fact you can see this by immunofluorescence, and this is just a quantification of the number of positive cells that you see over the time course. So this suggests that perhaps these cells are undergoing some sort of neuronal type of differentiation. And I'll say in data which we're writing up right now, we see the same type of effect when we re-express BRG1. We see the same phenotype, same gene expression pattern. And our collaborators in British Columbia have also done proteomics, and they can see the same proteins go up at the protein level. So why would SCOOTs be showing a neuronal differentiation? And this is where it becomes, I think, really interesting. So I mentioned we don't know the cell of origin. We do know this is a tumor that is extremely rare, and we have to understand both why it's rare and why is it only in these ovarian tumors. So if you go back to the old Vogelstein, is it this? You have a normal ovary and you get SMARCA4 mutation and SMARCA2 silencing, and boom, you have a tumor. So it's essentially almost like a 2-step or 1-step model. This seems like very naive to me, and I don't think that is the case. So we need something a little better. And what we like is the idea that the tissue that these cells arise from are already silenced for SMARCA2, because we do know that SMARCA2 becomes expressed later in embryonic development than SMARCA4. SMARCA4 knockout mice are embryonic lethal, SMARCA2 are fine. And we think it's because for the SMARCA2 knockouts, there's still SMARCA4 to compensate. For the SMARCA4 knockouts, there's no SMARCA2 to compensate. Again, get a deletion. I've shown you that we show that this happens. I didn't talk about this. We have a nice story about topo2A being part of the SWUSNF complex, and then it becomes less active in the absence of SWUSNF. And then also the preliminary ATAC-seq data supports the idea that you get more fluctuations in nucleosome positioning, allowing altered accessibility to transcription factors or to enhancers, and that may drive the development of this tumor. But why are these still so rare? This shouldn't be that rare of an event now. And we like the idea that perhaps these tumors are actually coming from a tumor type that already is very rare, such as a yolk sac tumor or immature teratoma. And perhaps these are tumors where there's already silencing in the cell of origin for these tumors, and then you can go through this process for tumor development. Do we have any evidence for this? Sort of. So what's the difference between immature and mature teratomas? Immature teratomas have immature neuroepithelium. And interestingly, there are several cases of SCOOHT tumors that we have where sort of off in the middle is still a section of immature teratoma within the tumor. If you look at, we looked at 3 or 4 immature teratomas now. Interestingly, I'm not showing all the other tissues, but all the different types of differentiating tissues that you see, all are positive for both SMARCA4 and SMARCA2. But this immature neuroepithelium is silenced for SMARCA2. So if this was the cell of origin, then you're already dealing with a rather rare immature teratoma, and on top of that, you'd have to have an inactivating mutation in SMARCA4 and loss of heterozygosity. So this might explain why these are such a rare tumor type. And then finally, as I said, there's several groups, especially Will Folk's group at McGill in Montreal, that are pushing the idea that this is just another extrarenal rhabdoid tumor. And they're actually calling it MRO, malignant rhabdoid of the ovary. And there are multiple features that are shared by these tumors. There's no other mutations that occur in SCOOHTs other than SMARCA4 or in rhabdoids other than SMARCB1. I already showed you there's a few copy number changes. P53 signaling pathway appears to be tact and functional in these tumors. They both have poor prognoses. They both show epigenetic silencing of the other ATPase subunit. And interestingly, 1 to 2% of SCOOHT tumors actually don't have SMARCA4 mutations. They have SMARCB2. And the same, and opposite with malignant rhabdoid tumors, 1 to 2% of SMARCA4 mutations. And this is just an example of a SCOOHT tumor that there's a typical one that's positive for SNF5, negative for SMARCA4, but here's another tumor that actually is negative for SNF5. And in these tumors, you can actually see the typical rhabdoid tumor cell with the whorls and intermediate filaments and the nucleus being shoved off to the side. I'm still, based on the P16 and some of the other data that we have, such as the gene expression, that these probably are distinct and probably do come from different cell types. But I want to point out this very interesting cancer genetics question, which is we have about 35% of SCOOHTs are familial germline mutations. And as you can see, these families are getting SCOOHTs, and maybe there's some evidence for anticipation, not entirely sure yet. But even the youngest patients here are 11, 12 years old. Here is a malignant rhabdoid family that also has a germline SMARCA4 mutation. These children all get malignant rhabdoid tumors, kidney or brain, at 7 and 8 months. So why do all these families not get rhabdoid tumors? They have the same type of inactivating mutation, yet the majority of these families bypass rhabdoid tumor development. And I think this brings up one of the really interesting questions in terms of the specificity, but also there's clearly has to be some genetic background or epigenetic background differences that account for this, and this is one of the things we're very much interested in. The other thing which I'm going to point out is that in general, rhabdoid tumors, and we're in the process of sequencing about 17 primary SCOOHTs, have the lowest levels of mutations that are seen in any of these tumors. So this is looking at the epigenetic load, where the top 5 mutations are epigenetic genes. And a lot of pediatric cancers fall here. And then, for example, here's melanoma. I believe this is head and neck and lung cancer. These all have very high background mutation levels, and none of them have epigenetic mutations, other than 10% of lung that have SMARCA4. And interestingly, Clear cell renal cell carcinoma has the mutation in PBRM1, another SNF complex member. AML has the mutations in the EZH2 gene. So my take-home messages are, number one, this is a very frequent event. A lot of the mutations are missense mutations of unknown function or impact at this point, and there's a lot of groups working on that. We still haven't resolved exactly how contribution of this complex to cancer occurs. We have been carrying out mass spec studies in the SCOOT cell lines, and we still find BRG1 peptides in those cell lines. Interestingly, it's a gene of 35 exons. You could easily imagine that you could splice out some of those mutations and have a truncated protein that may have some residual function. And this has always been one of the interesting questions I've gotten over the years from audiences such as you. Can a cell truly be SWUSNF complex negative? Is this not a complex that's so fundamental to regulation of gene transcription, replication, and DNA repair that cells actually would not be viable? And we're trying to address some of this by using CRISPR to try to get a complete knockout of the mutated gene in these cell lines and see if they're capable of growing. This is something which we are trying to put together a group to start looking at. I think it has something to do with where in development these mutations occur. I think in the case of SNF5, it's coming from a stem cell population, and that there's only a very narrow window in development when cells will tolerate loss of SNF5 and what it does to the complex. For clear cell ovarian tumors where there are ARID1A loss, again, it probably has something to do during the differentiation of the precursor cell, where there's, again, a narrow window where you could lose the complex. I mentioned already the EZH2 inhibitor story. We also have been looking at, there's changes in enhancers throughout the genome in the absence of SWUSNF, and we're looking at drugs that target enhancers. We have a paper that we're getting ready to submit where we've also been able to show that HDAC inhibitors work very well on these SCOOT cell lines. And finally, unlike most of the adult cancers, I think most pediatric cancers are really driven by an epigenetic instability. The selection for cells that move on to the next step and progression. And this is probably true for metastasis in general. Despite what you said about metastasis suppressor genes, I still think the majority of tumor progression is due to epigenetic changes. And having a tumor that is adept at making those changes probably makes it one of the reasons it's a much more aggressive tumor. And finally, I have a large multi-PI R01 on, and these studies are a combination of all these groups. These are the people in my lab. Krista Orlando is my very talented graduate student. David Huntsman is a GU pathologist at the University of British Columbia and is focused a lot on ovarian cancer development. And Jeff Trent is a director of TGen in Phoenix. He's driving a lot of the translational studies. And this is me from a few months ago, where we were looking at, we have meetings every 6 months. This was the one in North Carolina. And we're driving the studies that look at the epigenome changes. And this is actually Crystal. And Peter Jones and his graduate student, Ron Johnny, were responsible for the SNF5 data that I showed. And I'm happy to answer any questions.
Yeah, so it's interesting how you talked about the relationship between SWUSNF and PRC2. So mutation in SWUSNF complex, you unleash PRC2 activity, and that drives cancer. But some types of cancer are actually caused by loss of mutations in PRC2. So it makes me wonder if there are ever SWUSNF gain-of-function mutations, or in some cancers, SWUSNF makes a positive role in tumor development, and what's the difference between these two types of cancer?
So that actually was a question I gave to pathology graduate students in the graduate course a few years ago. How can EZH2 be both a positive and negative driver? Not remember what the answer. No. I'd say a couple things. One is, God, I hate getting old. It was there a minute ago. So one of the issues is that yes, and this is Sigal Kadosh's work. She had a follow-up paper recently that, again, the fusion protein, the synovial sarcomas, displaces SNF5 from the complex. And she can show that the resulting complex actually has a different set of targets. It goes to different places in the genome. So that is one example. And then this paper that's coming out shows that the mutations in BRG1, actually, the missense mutations, affect how the subunit hits DNA, where it binds DNA, in the groove, and actually affects, it again, alters where the complex goes in the genome. And most of the groups that have published so far have focused on the idea that you are affecting enhancers or super enhancers. There's some disagreement between Charlie Roberts' group and Sigal Kadosh's group about which set of enhancers. We still see a lot of changes in promoters, but enhancers loop around the promoter side. That hasn't been sorted out yet. But yes, there clearly are gain of function. And where you lose EZH2, I'm trying to remember this because I was just looking at this recently, there's clearly a different effect than the gain in terms of, if you think about it, if losing SWUSNF complex now causes repression of certain genes that are normally, say, tumor suppressors like P16, then losing EZH2 may allow expression of genes that are oncogenes. So you can make a model for both, and I think we're all very interested in terms of what are gain of function mutations. What we've shown with SNF5 is that when you lose SNF5, the complex, it's our hands and Charlie's hands basically falls apart. Gene expression levels don't change, it just becomes unstable. Charlie's also shown, and Kadosh has shown, that now the complex is targeting different sites in the absence of the residual complexes. So this is an evolving and complicated story at this point. Yes?
Enjoyed your talk. In the chromatin remodeling, there are a lot of other proteins that come into play that bind to the linker region and so on. And I'm wondering if the mutations in the various subunits influence the binding of those proteins. I mean, I'm thinking more of non-histone type proteins.
One of the issues right now, setting aside histone modifiers, is that this is an extremely sticky complex, and when we do mass spec studies, you can pull down almost anything. And if you look in the literature, it says, and I'm actually on one of the first papers, it says this, that the SWUSNF complex interacts directly with RB, it affects RB function. And there's a paper that says it interacts with P53, paper interacts with LKB1, with P16. Because quite frankly, every time you pull it down, because it's in chromatin, you're pulling down a lot of other proteins, and it's hard to tell exactly what's real. The only thing that we see consistently in multiple pull downs with 4 or 5 different complex antibodies against 4 or 5 different complex members is topoisomerase 2A. And there's clearly a biological correlate we can see to that. The original model was that, it affects the ability of RB to suppress gene function, and in the absence of SWUSNF, RB no longer turns off P16 and you get cell growth. It's still possible, but we're not clear. I mean, there's bromodomains in several of the proteins, including SMARCA4 and SMARCA2. They recognize acetylated histones. There's clearly some important interaction with the histones. That's one of the things we'll be looking at with our collaborators in British Columbia. They have an expert on ChIP-seq for the histone modifications. it would help if we knew what the cell of origin was so we could do some comparisons to see what the differences are, and we just don't have that right now.
All right, thank you very much.